Moxifloxacin is used to treat certain infections such as pneumonia, bronchitis, and sinus, skin, and abdominal infections caused by bacteria. Moxifloxacin is in a class of antibiotics called fluoroquinolones. It works by killing the bacteria that causes infections. Moxifloxacin comes as a tablet to take by the mouth. It is usually taken with or without food once a day for 5 to 21 days around the same time each day. Moxifloxacin is also used to treat tuberculosis and endocarditis when other medications cannot be used. Respiratory tract infections still account for causing significant diseases and death today. Moxifloxacin was developed to cure these infections since the bacteria grew resistant to the old antibiotics used to treat the same thing, such as penicillin. As you can see, during the late 70s to early 80s, the effectiveness of these medications were high. However, over time during the late 90s, bacteria grew resistant and these antibiotics became significantly less effective. Over time, knowledge of quinolone structure activity has increased, providing for a new and improved antibiotic, moxifloxacin. It calls for excellent gram-negative coverage plus enhanced gram-positive, atypical, and anaerobe activity. The chemical formula for moxifloxacin is C21H24FN304. Moxifloxacin is characterized by a fluoroquinolone structure similar to that of other antibiotics in the same class. The left side consists of methoxyquinolonic acid, difluorocyclopropyl, and dihydromolecules. The H3CO was added to minimize development of resistance and enhance anaerobic activity. The part circled on the left was added to minimize efflux and enhance gram-positive activity. This chart shows the steady-state plasma concentrations of moxifloxacin obtained with once daily dosing of 400 milligrams.
Topoisomerase II and topoisomerase IV are essential enzymes, which enable bacteria to replicate successfully by allowing the long helical strands of DNA to separate and unwind. Quinolones inhibit bacterial DNA replication by binding to the topoisomerase II complex above the replication fork. This prevents the strands from unwinding, stopping the processes of replication, transcription, and repair, and ultimately causes bacterial cell death. Occasionally, a mutation within a bacterial cell DNA can cause resistance to the older quinolones. The resistant cell successfully replicates, and the two daughter cells can now both carry the mutation, which makes them resistant to the quinolone. The chances of this occurring are about 1 in a million. Moxifloxacin is different to the older fluoroquinolones. The addition of the new methoxy group at position 8 helps moxifloxacin to simultaneously inhibit two targets. Not only topoisomerase II as before, but also topoisomerase IV, which, among other things, controls daughter cell DNA separation. Even if the bacteria are resistant to topoisomerase II inhibition, and the cell continues to function normally and attempts to replicate, moxifloxacin can bind to topoisomerase IV and inhibit daughter cell DNA separation. This causes both daughter cells to die, ensuring that the mutation cannot spread any further. To become resistant to moxifloxacin, the bacteria would have to carry two mutations. The chances of this occurring are minuscule, about 1 in a billion. By being active against two target sites, moxifloxacin is less likely to select for resistance.
In this animation, we demonstrate the biology of DNA replication, leading to bacterial cell division in a gram-positive bacterium, such as S. pneumoniae. The DNA is shown as a circular double strand within the bacterial cell. Like the DNA of all living organisms, it contains the unique genetic code for all of the proteins required for bacterial survival. Bacteria replicate by a process known as binary fission, whereby one bacterium separates into two new daughter cells. However, before this can occur, the bacterium must make an identical copy of its complete circular DNA. DNA replication requires that the two strands of DNA separate so that the genetic code of the bacterium can be read and a new complementary strand can be created for each of the original strands. To accomplish this, various enzymes, known as helicases, break the hydrogen bonds between the bases in the two DNA strands, unwind the strands from each other, and stabilize the exposed single strands, preventing them from joining back together. The points at which the two strands of DNA separate to allow replication of DNA are known as replication forks. The enzyme's DNA polymerase then moves along each strand of DNA behind each replication fork, synthesizing new DNA strands, in red, complementary to the original ones. As the replication forks move forward, positive superhelical twists in the DNA begin to accumulate ahead of them. In order for DNA replication to continue, these superhelical twists must be removed. The bacterial enzyme, DNA gyrase, which is also known as topoisomerase II, is responsible for removing the positive superhelical twists so that DNA replications can proceed. DNA gyrase is an essential bacterial enzyme composed of 2A and 2B subunits, which are products of the GYRA and GYRB genes. This enzyme has other important functions which affect the initiation of DNA replication and transcription of many genes. With the combined involvement of these enzymes, an entire duplicate copy of the bacterial genome is produced as the two replication forks move in opposite directions around the circular DNA genome. Eventually, as the two replication forks meet, two new complete chromosomes have been made, each consisting of one old and one new strand of DNA. This is referred to as semi-conservative replication. In order to allow the two new
Moxifloxacin is developed by Bayer AG and is marketed worldwide under the brand names Avalox and Avalon for oral treatment. It is available as a tablet or eye drop and is available at most drugstores.